Welcome back viewers to my channel. In this video we will be discussing about the primary and the higher order structure of protein. Now we know that proteins are unbranched polymer of amino acids, and these proteins are synthesized only from 22 standard amino acids. Now these proteins have four different levels of structural organization. They are primary structure, secondary structure, tertiary structure, and quaternary structure. So let us discuss in depth about the structure of protein. Now, primary structure of protein is nothing but a linear sequence of amino acid residue joined by a peptide bond. Here you see in this figure, these are amino acid residue, and this amino acid residue are joined by a peptide bond. And the structure formed here is the primary structure of protein. As I have said in the earlier video, the amino acid residue containing the amino group will be known as N-terminal amino acid residue, and the amino acid residue containing the carboxyl group will be known as C-terminal amino acid residue. So, primary structure of protein is a linear sequence of amino acid that is joined by a peptide bond. Now we know that linear sequence of amino acids gives the primary structure of protein. Now this linear polypeptide chain, when folded or takes into something like coil structure, it gives the secondary structure of protein. And the most common secondary structure of protein known are alpha helix and beta pleated sheets. You see here in this figure, this is the structure of alpha helix, and here it is the structure of beta pleated sheets. And this structure are stabilized by bond known as hydrogen bond. And this hydrogen bond will be forming between the carbonyl oxygen and the amide hydrogen of the polypeptide backbone. So when a linear polypeptide chains fold into a specific coil structure, it gives the secondary structure of the protein. Now let us discuss in depth about alpha helix. Alpha helix is a rod-like structure, and its screw ends can be right-handed as well as left-handed. In right-handed, you will see that the chain will be rotating in clockwise direction with respect to the axis, whereas in left-handed, you will see that the chain will be rotating in counterclockwise direction with respect to the axis. Here it is the structure of right-handed alpha helix. There will be amino acid residue, something like that. And in alpha helix, you will see that the complete one turn will contain 3.6 amino acid residue, and the length of one complete turn will be 0.54 nanometer, and the distance between two adjacent amino acid residue will be 0.15 nanometer. Now, since we know that secondary structure is stabilized by hydrogen bond, alpha helix structure is stabilized by intracell hydrogen bond, and this hydrogen bond will be forming between the CO group of each amino acid and the NH group of the amino acid residue that is situated 4 residue ahead. You see here in this structure, this is alpha carbon of first amino acid residue, second amino acid residue, third amino acid residue, fourth amino acid residue, fifth amino acid residue, and the sixth amino acid residue. Now the carbonyl oxygen of the first amino acid residue will be forming hydrogen bond with the amide hydrogen that is situated 4 residue ahead. Similarly, the carbonyl oxygen of the second amino acid residue will be forming hydrogen bond with the amide hydrogen that is situated 4 residue ahead. Now you see that not all amino acid will form alpha helix structure. There are some amino acid which cannot form alpha helix structure, and they are glycine and proline. Now, since we know that glycine has H atom as its R side chain, therefore this H atom will compete with the main chain atom to form hydrogen bond, and because of this, glycine cannot form alpha helix structure. And in proline, we know that both alpha carbon as well as the amino group is bonded to the R side chain. Therefore, because of lack of amino group, this proline cannot form hydrogen bond, and because of this, proline cannot form alpha helix structure, and therefore glycine and proline is known as helix breaker. Now another most common secondary structure is the beta pleated sheet, and this beta pleated sheet is formed when two or more polypeptide chain is arranged or lined side by side to form the beta pleated sheet. But unlike the alpha helix, which has a helical structure, in beta-pleated sheet the beta strand is fully extended and the distance between the adjacent amino acid residue is 0.35 nanometer, whereas in alpha helix it was 0.15 nanometer. Now beta-pleated sheet is stabilized by intersen hydrogen bond that will be forming between the polypeptide backbone H and the carbonyl group of the adjacent strand. Here you see in this structure, these are beta strand. And since this beta strand is arranged in opposite direction, therefore it is known as anti-parallel beta-pleated sheet. Similarly, when this beta strand will be arranged in the same direction, it will be known as parallel beta-pleated sheet. Now, the hydrogen bond will be forming between the carbonyl oxygen and the amide hydrogen of the adjacent strand. Now, tertiary structure is unique three-dimensional structure of a protein that is formed due to side chain interaction in their primary structure. As you see here in this figure, there is a disulfide bridge formed between the side chain and ionic bond in the side chain, hydrophobic interaction as well as hydrogen bond. Now this different type of side chain interaction will give or form the 3D structure of protein, that is the tertiary structure of protein. And because of this, tertiary structure is stabilized by both covalent, that is disulfide bridges, and non-covalent bond, that is hydrophobic interaction, hydrogen bond and ionic bond. Now, when two or more polypeptide chains are held together by non-covalent bond, it gives the quaternary structure of protein. These are polypeptide chain, and these polypeptide chain are held together by non-covalent bond like electrostatic interaction, hydrogen bond, and hydrophobic bonds. And the structure formed is known as quaternary structure of protein. And the best example of the quaternary structure is hemoglobin, because it contains 4 polypeptide chain held together by non-covalent bond. So when two or more polypeptide chain held together by non-covalent bonds to give a functional protein, it forms the quaternary structure of protein. So that's all for this video, and if this video was helpful for you all, please like and subscribe it. Thank you.